So this is a very interesting conference. It's so heterogeneous in terms of the applications that are described, and I really wonder if we are talking to each other or talking to ourselves. First of all, because big data is different when you have little money or you have a lot of money. So scientific applications, some of the talks touch on them, that have a lot of data but no funds for buying very expensive systems are very different than, say, what is done at Facebook or eBay. A second point is that I would like to make before I start talking is that also I used to be a computer scientist. I converted to more of an application person 10 years ago. When you hear so much about research, I was 30 years of conferences and talks about what is the future of database management systems. And they are kind of looking at in a direction that may not necessarily meet the needs of what is actually happening in the scientific data management world. So I will try to touch on these points as I go through my talk. And also clarify a little bit what this genomics area is. What we pretty much have, it's a large data warehouse and a bunch of analysis tools. There are tens of them. All of this analysis is done interactively. And this is a very popular system. It's 10 years old today, has lots of users worldwide. The numbers that you see on these slides are registered users. These are people who actually come to the system, bring their own data, integrate it with the data that is already in the system, and conduct microbial genome analysis. The increase of the number of users is, and the number of students that are using the system for educational purposes is pretty much for us a sign of its value. So what I'm going to talk about is a little bit clarifying what genomics means, what the genome data is all about, the sources, integration, and the maintenance of the data warehouse itself, how the analysis tools look like. So the data warehouse and all this infrastructure really serves one purpose, and that is providing the analytical capabilities for scientists to do their work. And we'll end with some overview of the database technology. My colleague Amy Chen is going to talk about it, and pretty much is explaining what can we do with existing resources to provide science. Science is not going to stop and wait for the grand solution to come. So genome data. We are working with the Joint Genome Institute. It's one of the largest sequencing facilities in the world. They sequence a lot of microbes from various organisms. It's one of the big sequencing centers. You see on this slide what other centers are. And what the revolution in the sequencing world was that the price of sequencing has dropped dramatically, and therefore the amount of sequence data that has been generated, that keeps being generating, is rising exponentially. So this is the quote-unquote big in this particular world. There are 4 types of genomes that are sequenced: plants, fungi, microbes, and metagenomes. I am focusing on microbes and metagenomes. That is where the numbers of the organ is sequenced, and the size of the data sets is very high. And so that we are clear about what we are talking, genomes, you may know, are associated with organisms, such as humans, have genes. Genes are associated with functions, and they usually come in the context of pathways. So no single gene works alone. No single function is relevant unless it is put in this context. Metagenome, which is a newer concept, is pretty much the aggregate genome of all the genomes in a community of organisms, such as the microbial community, the microbiomes that we have in our gut and in all parts of our body. So pretty much what is happening is once an organism is sequenced, It starts with raw sequences, goes through taking those raw sequences, putting them together, that is called assembled data, and then going through a process that is called annotation. So pretty much looking at the sequence of data and figuring out the regions that are genes that may actually encode the function, and then figuring out what that gene may do. And then is gene expression. So actually, gen-based Z genomics application was talking about expressed genes, not genes themselves and their functions. So this is a third kind of aspect of it. So you have data transformation going from raw sequences to assembled data to annotated genes, and then data characterization, taking genes and figuring out what they do and under what conditions they do these things, which is the gene expression part. All this is then integrated. So for many years, 10, 15 years ago, individual organisms were sequenced because sequencing was expensive, and they were analyzed one by one. That doesn't work anymore. First of all, the analysis is very tedious and actually yields very few results. So what is currently done is all the data that is available is integrated, put together. What integration means is looking at correlations between various genomes. So gene in one organism, how does it relate to a gene in another organism? This is pretty much what it is done. So you take and figure out all these correlations. What makes this very difficult is that this process of data interpretation, the data transformation, integration, and characterization, it's an iterative process. It does not stop. So you go with one data set, go through all these steps of interpretation, integration, you do analysis, figure out that something doesn't look quite right, and you can go all the way back to resequencing. So this is another big source of data. So this is a big picture that one needs to keep in mind. It's not what is at the end, not what a database looks like, but actually how the database lives. It's live of getting data in and continuously, iteratively improving its quality. Okay, data are distributed. We are actually working with NERSC, so we have access to a supercomputer. Supercomputers help a lot with the data transformation and integration, so they can do a lot of computations. They do not help at all with the actual data warehouse and supporting analysis. So the sources of the data warehouse. There are public genomes. There are functional resources. No need to get into the details of what they mean. What really matters is that there is computation once the data gets into the warehouse. And what really matters are these numbers of the growth of the amount of data. So this is what we are tracking pretty much on a daily basis of how many new genomes are we getting from the public domain, again, because we want to have all the universe of genomes and as many metagenomes as we can in order to improve the quality and the power of the comparative analysis that is conducted by scientists. So these numbers show you the growth over the past several years, since 2009, when the price of sequencing has started to drop dramatically. And the objects that people are interested in are individual genes. That is why we count how many genes we have across individual genomes and how many genes do we have across microbial communities, and you see that there is a huge difference. This is how currently the analysis is done. It's a gene-centric analysis when you go from tens of thousands, hundreds of thousands to millions and then billions of genes. That is where the challenge is. This is the context of the analysis, and this is again these are screenshots from the first, the first page of our system. When you get in, the first thing that the scientists will do is look at how many genomes are there and what is the depth and the breadth of the annotation, how much information is available. So let me now go to the analysis tools briefly to give you a sense of how analysis is conducted. There is a data warehouse that all the data are integrated, all genome metagenomes goes into one big pot, and around it there are a number of data marts that looks at subsets of the data. So there is difference between genome analysis. Some people want just to focus on isolate genomes. Some people want to focus on metagenomes. Then you have the slice of people who look at isolate genomes or metagenomes but want to do expert review of this data. What that means is looking at trying to find what is missing in the data, or if something looks incorrect, and fill in the gaps and correct the errors. And that is another slice that goes across genome analysis and metagenome analysis. And then there are a number of special purpose data marts that focus on special type of genomic analysis, such as a human microbiome project that looks at the human microbiome. This is a data mart that focuses on that type of data, and there is a data mart for education purposes. This is pretty much a list of all the tools that are available. There are two types of analysis, so it's pretty straightforward. But what I want to actually tell you is that it's not that complicated. It is pretty much looking at what is there, data exploration, and the comparative analysis, which is comparing things. So if you look at it, is pretty much like drilling down operations and rolling up. So it's really genomics at its basis, and I would say 75% of the analysis is of this nature. You look at a multi-dimensional space. The key 3 dimensions are genomes, genes, and functions. And you start by looking at functions. You do some summarization of where these functions appear across data sets, across individual genome data sets or metagenome data sets. And then once you do that, then you start drilling down and looking at picking up organisms of interest, several of them, and you start comparing them, kind of focusing the analysis on subsets of genes and or subsets of functions. So it's pretty straightforward from this point of view. Therefore, the question then is, what is a database technology that could support it? And the answer is straightforward. If we would have enough funds, we could buy an expensive enough and powerful enough system to provide support for it. But we don't. That is a problem that we are facing, and I believe that most of the scientific applications are in the same face the same challenges. When we started in about 2006, our data sets were pretty small. We had an Oracle license, was perfectly adequate for what we needed, for that type of analysis that have not changed over the years, by the way. But again, our context is a budget is available for a limited Oracle license. Once we saw the data growth, we simply could not afford buying a bigger Oracle that would have cost us much more money. We had the limited resources, only one database expert to look and try out the various systems. All of them promise you a lot. In reality, they actually provide you not quite what is promised. And we had a live system, so we couldn't just build another system and try out another technology over and over again, because as you saw, it's a live system that is growing very rapidly, and scientists need to get access to it and do their work day in, day out. So over the years, what we have done, we pretty much had the scavenger approach. Take the Oracle system, keep it as it is with the limited license we have, and add on it other systems, try things out. We tried them for a while, threw them away, tried something else. We have some files. We use some zipped files. It worked. We used SQLite. We tried all kinds of other systems. We are using something that was developed at Lawrence Berkeley National Lab for physics application called FastBit, and it does provide support for one operation out of the tens of operations we have. So we have this collection of ad hoc technologies to provide support for the type of analysis that are needed. So these are the system layers. Pretty much the visualization is straightforward, and scientists actually know very well what type of visualizations they need. The operations are not very complicated. It's just how to provide support for interactive type of analysis, pretty much with what type of system. So we experimented when we knew that we cannot sustain the growth with Oracle. We experimented with Hadoop, HBase, SciDB, Hive, none of which worked out, and this is what we have now. We have a combination of files, FastBit, and SQLite, and I will pass on now to Amy Chen, who is going to explain how we came up with this.
As Victor described, we have a 3-layer of system. At the top is the UI, then we have analysis layer, and at the bottom we have the database. We start with Oracle database. So I would like to say a bit more about the UI. When we start to implement IMG in 2005, we have two kinds of user interface. We have web UI and we have Java application. But biologists, the majority of them prefer to have the web UI because it's easy for them to use. Just go to a URL, you can start doing your analysis. You don't need to download software, install, and things. Therefore, we are also limited to the limitation of web browser, which means that it will time out in a couple minutes. So you cannot have a very slow analysis. Okay. Victor has already described that we are doing the 3-dimensional genome, gene, and function analysis. So it will be very natural to you to say, well, this is basically a data cube, so why don't we just use a data cube? It's simple and straightforward. Unfortunately, it does not work with our production environment because we have users continuously submit data to IMG. We do data loading every day or every week. Continuous data loading interferes with indexing and data retrieval. So you know that if you leave the index on, then the data loading will be very slow. If you disable the index and load the data in, then indexing takes forever. Back in like a couple years ago, when we were still sticking with Oracle database, it took us like 2 weeks to reindex the entire database. So it's just not feasible. And also, Victor mentioned that we have those resequencing and resubmit going on. Because when biologists look at the raw sequence, ACDT, they just cannot make sense out of data. They need to submit their data to IMG, and through gene coding annotation, then they use the web UI to look at data. It's not uncommon for them to find out there's contamination in their data, like human, dog genomes, or E. coli, MD1. So what they did is that they decontaminate those genomes and resubmit into IMG. And this sequence can go through multiple times. And there's also a couple of times that some biologists found out they actually submit the wrong data files in, so they have to resubmit the data. And when there's new data set going on, usually they want to have the old data set removed. Otherwise, it's not only confusing, but also when you do the similarity analysis, you will find out that the data set that most similar to your data set is your old data set. And we know that a lot of existing data warehouse, data cube implementation, they are not good to deal with obsolete data, the deletion of the data. And many of them will suggest that, well, why don't you just mark them obsolete, but don't actually delete the data. It's work fine only to some limit, because as I mentioned, a data set may be resubmitted to 5 or 6 times. If you don't physically delete them, then your system may ends up to have more obsolete data than the real data. So it's really bad for your storage. And also, a lot of analyses are based on the count of the function in a genome and the comparison of this count. And all of these counts are kind of predetermined when your data set is loaded into IMG. So supposedly, if you have a genome which have 5,000 genes associated with this cog X, no matter how many times you count, you get 5,000. Therefore, if you use this data warehouse and you keep doing the rolling up, it's kind of like a waste of time. But the major problem of using new technology is the budget. We cannot find any technology that's within our budget that can keep up with our data growth. We reached the 1 billion gene data mark at around the end of 2011. Now we have close to 20 billion genes, and it's like 20 times larger in less than 2 years of time. Okay, so what did we do? We still have Oracle database. So what we did is that we put our genome and our gene function count, those the pre-computed roll-up count in the Oracle database, which is fine because the size of data is pretty small. But for the individual genes in the metagenome, we partition them and put them in a different data set, and we use the open source database to store that. Our current favorite is SQLite database. And the N here is different for every metagenome because metagenome is the genome community. So we have large and small metagenome. For some small metagenome, we may just divide data set into 2 or 3. But for large, you may divide into several hundreds or thousands of them. So using this implementation, we successfully overcome a couple of problems. The most important one is the budget problem, because we use existing Oracle database and then we use open source database, so we can still keep extending within our budget. And also because metagenome data now are partitioned into different data space, so data loading does not interfere with user querying the existing data. And it's also easy for us to delete the data set because they're in different data space. We can just trash a portion of that one. And our function count is efficiently, can be efficiently used for those analyses that only need the count. But of course, nothing is perfect. The problem with this approach is that, I think Victor kind of quickly went through his slide. In IMG, we have something called workspace. Users will be able to save their genome and genes and scaffold into a set. And in this set, they can add new data in, or they can do some set operation, union, set difference, intersection. And for those, we just cannot pre-compute. Therefore, if users want to analyze those kind of data, it will be very slow. And our workaround here is that we use background computation using a message system, which use RabbitMQ, and this is a mechanism provided by nurse staff to help us. So if users want to do analysis on a workspace, gene set, or scaffold set in IMG UI, we let them submit a job. They compute the background, and after the job is done, they can use that to check the result. And Victor also mentioned the FastBit. We use this in one of the important operations. This is the cassette search. So you can see here, each of the color, the arrowhead here is a gene, and it's a line here shows how genes line up. So why is this important? Because from biological point of view, usually a gene related to the same function are lined close together in this one. So we want to know how they are related and how the functionality of this gene. And in the relational database, of course, we store gene and function in each row. So if you want to do analysis using relational database, it's very slow. So it can take tens of minutes and it's not scalable. What we did, we used the FastBit is developed by John Wu and Lawrence Berkeley National Lab. It is basically a memory-based bitmap. So when we load this data in, and so it basically shows which cassette has genes on which function, and because it's in memory, so it's very fast. Richard, do you want to look? Okay. So the summary is that in IMG system support microbial genome and metagenome comparative analysis, and it's serve a large community of the user. And scalability has remained a major challenge, basically because of our budget limitation. And currently we are using Oracle, SQLite, FastBit, and some files. We use files to store sequence data and or pathway image data. In our futures there, we need to keep exploring technology because there's always new sequencing technology come in, and data just, we have more and more data come in. And we have tried experiments with HBase, Hive, and CyDB. They are not successful. The problem with HBase and Hive is because it took too long to do data loading. The problem with CyDB is because it's a couple of problems I already described in the data cube. It's basically indexing and data loading problem, and also we cannot do deletion. Another problem is that our gene actually have string type of ID, not integer type of ID. When we try to do redimensioning or reindexing using CyDB, we run out of memory. So this basically, we don't have a robust system based on our budget limitation. Okay, I'm done. People, do you want to take questions?